I was going to focus on, I was mentioning earlier that I called, by my title was supposed to be myeloid response to cancer, but in fact, what I'm going to describe in an inflammatory response. Most cancer, in fact, most injury, any type of injury lead to the enhanced production, release, and recruitment of myeloid cells, mostly monocyte and neutrophil, to inflame tissue. Now, it's very important that we think about that, is that really anything in the periphery, little burn, and likely also a small tumor lesions, as I'm going to show you, leads to that release of myeloid cells and myeloid cells from the bone marrow to the blood and the recruitment to the tissue. This is how we fight against injury. Now, among this myeloid compartment that is being mobilized, there are two cell types that we are very interested in. Macrophage and dendritic cells that were, this is especially that were introduced by Jonathan earlier. And these cells are quite unique in the immune system in that they can be mobilized in response to inflammation, mainly through a progeny. And I won't have time to talk about this, but they are also present, and this is at the tissue site prior to the injury. They are only the main immune cells that are constantly surveying, normal tissue. So they are the site of the lesions prior to any injury, and therefore they are the first one to respond to it through the release of inflammatory molecule that is going to shape that response to the injury. So we believe, for example, that release of myeloid cells that I discussed earlier is being released in response to cues that are induced at the lesional site. And these cells, because of their very sophisticated machinery that allow them to sense many different types of threat, they are going to contribute to really inducing that systemic response that is going to shape the response to injury. And it's very important to understand that, right? So when we think about a tumor that is hot or cold, it is possible that the signals of these two compartments that are always present prior to any type tumor lesion is contributing to the hotness or the coldness of the tumor. Now, there are uniqueness between these two compartments. Dendritic cells are uniquely able to migrate to lymphoid organ and structure in tumor lesions. As Jonathan mentioned, Jonathan insisted on that CCR7 molecule that allowed them to migrate, because with this molecule, they migrate to T-zone of lymphoid structure. And this is where they can really instruct T cells in a way that is really very unique to that compartment. It has all the molecules that are required to initiate a T cell response in a way that is very unique, even among, antigen-presenting cells. Macrophages, though, are much more primitive cells which are equipped to promote inflammation, like DC. If they sense something, they release an inflammatory molecule. But most of their equipment is directed towards tissue clearance, threat clearance of threat, microbe, damaged cells, but also tissue repair, right? And they do all of these. So they are a complex compartment, and at some point when in inflammatory lesion, they turn on a very suppressive program because they want to quiet things down. So initially they're inflammatory, and at some point they become suppressive because they want things to quiet down, right? And because so it is, we think about them quite differently. When we think about DC manipulating dendritic cells in cancer, we think mostly of harnessing their ability to modulate T cell responses. When we think about harnessing macrophages, we think mostly of harnessing potentially their ability to clear tumor cells, although I don't work on this in the lab, but many groups do. But we also think of reprogramming that suppressive pathway that is induced in tumor lesions. Okay, now there is, despite the realization that macrophage and DC have key role in tumor immunity, as Jonathan described, there is no or very few FDA-approved macrophage and DC targeted therapy for cancer. And we think that the development of this myeloid targeted therapy lagged behind because the exact molecular state that really hampered DC and MAC's ability to promote this anti-tumor immunity are poorly understood. So these extensive profiling, and we've now profiled many human tumors and millions of immune cells. And this extensive profiling, in fact, had revealed common pattern of myeloid response to tumor cues. And we have, in fact, strived to search for conserved program of these two cell types that I described to you, macrophage and dendritic cells, because this is what we study in the lab. And we search for programs that are conserved across tumor, because this is the low-hanging fruit. First, we want to know those that are shared across tumors, because we know that they exist, but also across species, so that will enable us to study causality, right? So this is what we did here. Here we profile, for example, and what we realized is that, in fact, there were much more commonality between so we profiled with single-cell RNA sequencing the cancer lesion and adjacent tissue that was free of tumor. And the first thing we realized is that, in fact, there were many more commonality between cancer across patients than between the adjacent tissue, suggesting that, in fact, what we were seeing is response to tumor cues, and the tumor cues across patients are often similar, right? They are like a cycle of cell death and proliferation, cycle of release of reactive oxidative species that, in fact, are going to give rise to a group of signals that are, in fact, shared across patients with the same tumor, but also across tumor. So so now we zoom in on Max, and I'm going to talk about macrophage the rest of this talk. So I'm going to zoom on using that single cell approach and ask, okay, what are really the molecular programs? So the way we do this is that we are going to look at cells, because this is done at the single cell level, that share the same transcriptional program. And we are going to group them in a group of cells that share this program. So we gate on a marker of macrophage, right? So marker of lineage. And then within this, we are going to look for heterogeneity. Okay. And this is what we call a macrophage cluster. So it belongs to the macrophage lineage, but within the same tumor, there is different clusters. So macrophages are going to differentiate into specific molecular program. And there is one program that became very interesting to us that was dominated by the TREM2 transcript that was present across many different tumors and expressed all these molecules here, at least 10 to 15 genes that were present, across many tumors that we've looked at. We verified, the expression of these TREM2 transcripts that dominate these clusters using an antibody we used to stain. This is a human non-small cell lung carcinoma. Now, this was quite interesting to us because when we identified these clusters, now many years ago, this cluster was found, in fact, initially in Alzheimer's lesions of patients in the brain. Okay. Now, this TREM2 is an immunoglobulin receptor that binds to phospholipids that are exposed, by apoptotic cells. It also binds lipoprotein, ApoE, and it also binds, that protein beta amyloid in Alzheimer's disease. And we know that these TREM2 signal to adapters, mainly DAP12, but probably also DAP10. Because we became quite intrigued by these. And in fact, that TREM2 variant have been associated with increased risk of Alzheimer's disease. And the TREM2 program was not only found in Alzheimer's, but also in many inflammatory injuries. So, for example, NASH, but also in other tumors, in mice and in humans, but also in fat tissue. Okay, so we realized that, in fact, what we are seeing is a response that was not specific to cancer, but maybe responding to some type of lipid sensing. Okay. So we went to mouse and asked whether that program was also present in macrophages that accumulate in mouse lung cancer lesions. Here, we injected mice with a cell line that was developed by Tyler Jacks many years ago, a lung cancer line that expressed a KRAS-activated mutation and P53 deletions. Mice develop adenocarcinoma, and we injected orthotopically. They developed these adenocarcinoma lesions in the lung. Then we use the same single cell profiling, and we look for this program, the TREM2 program. Okay, so we are going now to analyze these myeloid cells and cross-analyzing with the human finding. And then what we saw is that these programs that we identified, the TREM2 program with this 10 gene that I described earlier, were also present in this mice tumor. So that became quite exciting. exciting to us. So we said, okay, so since TREM2 is dominating this program, what if we first try to no, first things we did is let's try to recapitulate whether we can induce this TREM2 program in vitro. And because TREM2 was described as, these lipid sensors and potentially that were exposed, in apoptotic cells, what we did is we took a monocyte and then we exposed it to tumor debris, and then in the presence of different cytokine. And one cytokine that was really important here was IL-4. And then, as was described, in fact, this experiment was already described by Marco Colonna many years ago. Although I'm not sure he looked exactly at whether he fed cells with debris. We fed cells with debris. Marco had shown that monocytes that are exposed in the presence of IL-4 and GM-CSF can differentiate into cells that express TREM2. Here, what we did is we took a monocyte, we fed them with apoptotic cells, and then in the presence of IL-4 and CSF, I believe, and we saw part of the program being induced, ex vivo, suggesting that capture of cells in the certain cytokine milieu lead to that program that we identified both in mice and human tumors. Now we knock down this TREM2, only one transcript, and what we saw is part of the program was strongly reduced, suggesting indeed that uptake of this apoptotic debris induced TREM2 that will lead to this program that we identify in the clusters. Because when we deleted TREM2, that program was reduced. But in addition, what we saw is that we were able to reprogram macrophage into an immunogenic cellular compartment. And in fact, not only macrophages were more immunogenic, the DC in the vicinity of the tumor, where this is done now in vivo. Sorry, this was done in vitro here. Here now we go back in vivo, we knock down TREM2, and then we look at the macrophages. But these specific DC that do not express TREM2 were also more immunogenic because maybe DC were producing inflammatory cytokine in this new state where they lost TREM2. Now, what we also saw, then that there was a strong reduction of tumor progression in these mice in which TREM2 is deleted, the TREM2 is knocked out. And what was interesting to us is that this was associated also with increased accumulation of NK cells. Now, we often see a reduction of NK cell in most of the tumor that we look at, because compared to the adjacent tissue. And in fact, we had find similar results in our human lung cancer lesions or many of the tumor lesions that we've looked at. And we believe that this depletion of NK cells is very important for tumor to progress. So here we were potentially in the presence of a macrophage program that was contributing potentially at depleting these NK cells so that the tumor can progress. And maybe macrophages were doing it in order for, in thinking that they needed to repair the tissue, but what they were doing is allowing the tumor, these tumor lesions to continue to grow. So then what we did here, because the goal of this course is to think about therapeutic opportunities. So what we saw here is that macrophage can be reprogrammed through modulation of this TREM2 into an immunogenic phenotype. That when we did that, we potentially increased the accumulation of NK cells. And in fact, we saw that NK cells were also more functional. We now treat these mice jointly by this TREM2 antibody and this antibody that Lucas did to engage NK cells. So reduce this macrophage program that reduce NK cell function and at the same time increase NK cell effector function. And here we saw that indeed this combination was quite synergistic in reducing tumor growth, and this reduction in tumor growth would depend on NK cells. All right, and now when we go back to the human tumors, what we see indeed is there was an inverse correlation between the presence of this TREM2, the presence of macrophages and the presence of NK cells, suggesting indeed that, that this macrophage may have contributed to this NK cell depletion. And then we explored, exactly how this works, why, macrophage were depleting NK cell accumulation effector function. I don't have time to go into this, but to us that was quite interesting because then we started to understand the dynamic of this inflammatory component and how we can really harness it specifically to eliminate tumor. And these TREM2 macrophages were present mostly at the tumor site. So it's possible to really focus on this program with that focus on this program because it was not present in normal tissue. So this enables us to really to identify tumor-specific macrophages and potentially target them, reprogram them, and use them to really and use them to reprogram that inflammatory response and direct it toward tumor cells. Now, if I have time, yeah, I do have time. I'm going to talk about another program that was shared across different tumors. Okay, so here I described TREM2, and to us it's very important to really think about commonality of program, at least until we exhaust them, and then we will be focusing on programs that are specific to each different tumor. So now we are looking at, we continue our search of these different programs across different tumors. So we always look at a program across 3 to 4 tumors, and then we go back, the framework is, then we go back to mice. So here we ask, is there any other program that we should be focusing on? And we were surprised to find a molecular signature that was dominated by, or thought to be driven by IL-4 in both myeloid, in both macrophage and dendritic cell that infiltrated lung cancer and other human tumors. And that was surprising to us because this IL-4, which is part of this type 2 cytokine, which is known to dampen, in fact, type 1 immunity that is important to eliminate tumor cells, we had not realized, that this type 2 signature was dominant in tumor lesions. Okay, so we had seen that, but in human tumors, we then went to our mice lung cancer models, found exactly the same results, and we had blocked IL-4 a few years ago and saw that these, in fact, led to reduced tumor progression. So blocking IL-4 somehow was beneficial. So more recently, what we decided to do is to ask how this IL-4 really works in these tumor lesions. And we decided to block IL-4 at different stage. To block IL-4 receptor, because what we were seeing here is an IL-4 receptor signaling pathway. We were seeing that myeloid cells were responding to IL-4. And we found that IL-4 blockade was somehow beneficial to tumor progression. So we decided to remove genetically IL-4 receptor at different stage of myeloid differentiation. And we found that it was only when we deleted IL-4 receptor from early myeloid progenitors in the bone marrow that we were able to see a significant anti-tumor effect on the same lung cancer model that I described earlier to you. Okay? So if we delete this IL-4 receptor early during myeloid progression, we saw that there was a strong reduction of tumor progression. Okay, so they suggest that somehow these myeloid cells that were responding of IL-4 was contributing to tumor progression. So then we went down and asked what type of signals, really happen upon this IL-4 receptor deletion, what type of reprogramming of the tumor inflammatory component happened. And what we saw is that there was first a strong reduction of monocyte in the blood. Remember when I described this release of monocyte in response to peripheral injury that was reduced when IL-4 receptor is deleted. So that was interesting to us. We then saw also that the macrophage that were infiltrating the tumor lesions, including the TREM2 macrophage that I described to you, were much more immunogenic at the tumor site. Here, remember, the IL-4 receptor is deleted only in myeloid compartment, and we saw a strong accumulation of T cells and NK cells in the tumor lesions. Okay, so that was very interesting. So, and while we were doing this study, our colleague Brian Brown was developing a model to probe in a semi-agnostic manner the contribution of myeloid factors produced by tumor lesions on defining, that myeloid microenvironment of tumor. And he developed these fantastic models called PerturMap that will enable him, in fact, to develop a pooled CRISPR screen. And that CRISPR will each be attached to a tag that could be recognized, a tag that will, in fact, label both the nuclei or the cell membrane. So that will enable us to identify the tumor cells that receive each specific gene editing program. And he used this CRISPR screen, introduced it to different tumor lines, in this case, an ovarian cancer line, and then they convolute, right? So they convolute this relationship between the specific CRISPR and its myeloid microenvironment using spatial imaging. Okay, so this is what he does here. He introduced different CRISPR. Each clone of the tumor will have one specific CRISPR. The CRISPR has a tag, so you can recognize what CRISPR was introduced in what cell clone, and then he will then correlate the specific gene editing with the myeloid or the immune microenvironment or the inflammatory microenvironment of each clone. And what was very surprising from this early result that was published in fact 2 years ago, is that it seems that at least there was no contamination of microenvironment between one tumor to another, as if there was in fact discrete force that will shape and retain that microenvironment around the tumor line. But what was very surprising, well, but what we then did, because the results were going to come up in my lab, Brian decided to now delete this IL-4 add this IL-4 in his pooled CRISPR screen. And he did this experiment where he now introduced, injected the mice with this ovarian line that had different CRISPR, including that IL-4, which is the molecule of interest in this case, and then treated the mice with PD-1 blockade and looked for tumor that will regress more or tumor that will grow more. And what was very interesting is that the tumor that lack IL-4, that could potentially signal on the myeloid compartment, were in fact more susceptible to PD-1 blockade. This is all the CRISPR that he put in his tumor line. Here, in inner circle, the tumor are not treated, outer they are treated, and he showed that when there is no IL-4, they are more susceptible to PD-1 blockade. So then he did a tumor line that is now a single knockout, and in the single knockout, he removed IL-4, right? Only single knockout, no other CRISPR, and then ask whether this, in fact, changed tumor progression. Here, what he did, the tumor doesn't have IL-4, he treated with PD-1, he sees that he improved the tumor progression. Here, what he did is he removed the receptor now from the myeloid compartment, exactly like we did earlier in our lung cancer lesions, and then introduce his ovarian cancer line and treat mice with PD-1 blockade. And in this case, what he finds is that indeed, just by removing the IL-4 receptor from the myeloid compartment, these mice with ovarian cancer did much better in response to PD-1 blockade. Okay, so very similar results in a very unbiased manner. Us, what we did is we found it in human, right? We went to mice and found exactly the same signature. Then we deleted this IL-4 receptor from myeloid compartment. He was doing a pooled CRISPR screen. He deleted that IL-4 gene, so exactly the same phenotype, then made an IL-4 knockout. And then he went back and did exactly the same experiment where he deleted IL-4 receptor from the myeloid compartment and see a dramatic effect on tumor response to PD-1 blockade. I hope I'm explaining here that together, we were trying to go in to really utilize, unbiased profiling strategies to identify potential program of interest, right? And we were and together we arrived at the same finding that this IL-4 receptor signaling was detrimental to tumor immunity. Okay, now this is a very recent experiment, which is really a work in progress, where Jesse Bullman in my lab, a postdoctoral in the lab, in fact, he was working on colorectal cancer model and trying to identify myeloid target of colorectal cancer. So he did exactly the same experiment as Nelson or, in fact, Gurkan in Brian's lab, he introduced colorectal cancer line into mice in which we had deleted the IL-4 receptor from myeloid progenitors. And he saw that these had a dramatic effect on colorectal cancer growth in mice. Okay, so this led us really to all these results together to explore whether we could potentially block IL-4 receptor signaling in patients with cancer. Now, it happened that there is an antibody against IL-4 receptor, a blocking antibody against IL-4 receptor that we used, in fact, in millions of patients with allergy, mostly atopic dermatitis, but now the same molecule has been approved for severe asthma and has been approved also for COPD. And we know that this molecule is safe. Don't know whether I should go into this. I don't know whether the panel, the moderator can tell me whether I can continue or should I stop here? What do you think? Nicolas, Jean, what do you think? Should I stop here? Because we have 15 minutes, or should I finish?
You should continue.
You have 10 minutes.
I should continue? Okay. So, what we really became interested in, this realization that at least in part of our story, and I didn't detail that, and maybe we can discuss it during the discussion time, that IL-4, that when we deleted it from early myeloid progenitors. And at least in our lung cancer, we realized that IL-4 was being produced in the bone marrow by bone marrow granulocyte. This was not the case for the ovarian cancer where IL-4 was being produced at the tumor site. And it's possible that IL-4 is being produced at both at the tumor site and at the bone marrow site. But what we realized really is that the bone marrow was sensing the periphery, right? It was sensing the periphery. And this was, of course, obvious, right? Because we know, and we and others have seen that when you inject, a tumor orthotopically, and when you look at patients bearing tumor, we know that there is an increased release. We know there is an increased production because we see an expansion of hematopoietic progenitors, myeloid progenitor, granulocyte monocyte progenitor in the bone marrow in response to tumor, and this grows, in fact, this myeloid expansion is further amplified during tumor progression. And we see exactly the same thing in patients where there is an expansion of both early hematopoietic progenitors and myeloid progenitor, early hematopoietic and committed myeloid progenitors. Okay, so if there is expansion of myelopoiesis, it means that there are cues that are inducing systemic cues coming from the tumor that are leading to this expansion. And this is becoming a big focus of the lab, and this work is being led by Samarth Hegde, a postdoctoral fellow in the lab who will be soon, in fact, on the market. So what Samarth wanted to know is identify the epigenetic cues that are leading to this myeloid bias during tumor progression. So what he did is he purified myeloid cells, in fact, at different stages during myeloid differentiation, starting from early myeloid progenitors to circulating monocytes to macrophage in the tumor. And analyze them using this technique of ATAC sequencing. This ATAC or combined single-cell ATAC and RNA sequencing. This is a very expensive technology, but we are going to publish all that for you. Many of you won't have to do this type of study yourself, and you can just analyze the data that we will produce. So here, what we are trying to do is ATAC-seq allows you to see the part of the DNA that is now being bound, that is not bound by, that is the part of the chromatin that is open, suggesting that if the chromatin opens up, means that it's being targeted by a cue. Right? And then using different type of software, we are looking at regions, at potential binding motif of these open chromatin regions. So ATAC-seq allow you to sequence the open chromatin regions, and then using software analysis, we are going to hypothesize on potential binding motif that are binding these open chromatin regions. And we combine that with single-cell RNA sequencing that is going to infer potential transcription factors that are present in the same cell, that is maybe corresponds to this binding motif on open chromatin regions. And we did this in mice, and we did a similar exercise in patients, in lung cancer bearing patients. So here we didn't take hematopoietic progenitors, we only took circulating monocytes and macrophages that infiltrate the tumor. And now we are going to do the same thing. We do this cross-species analysis to look at open binding motifs that are shared between mice and humans. But first, we group this binding motif by class, by protein class. And what we saw is that in these myeloid progenitors, there was a very strong, in fact, expression of an integrated stress response, as if, in fact, myeloid progenitor, both in mice and humans, were responding to ROS, reactive oxidative stress, which we know are abundant in tumor, that had traveled, to the bone marrow. And we were quite intrigued, so we look at all their many molecules, but there is one molecule that was not poorly studied, at least in the context of myeloid biology, which is called NFE2L2 or NERF2. And this molecule is in fact known to really suppress inflammatory response, suppress type 1 interferon response and STIG response, which we know is important to promote innate immunity and in fact drive T-cell effector function. Right. So we said, well, okay, so this transcription factor is being induced maybe to protect. The reason why this NERF2 program is induced is to dampen inflammation, but also promote cell survival, right? And and we became interested in exploring whether this was contributing to myeloid suppression that we identified in tumor lesions. Okay, so bear with me here because I love this slide, but maybe difficult to explain. So here we said, okay, so we were interested in exploring the hit in early myeloid compartments, right? So here what we did is ask where that NERF2 signaling happens, right? Does it happen very early on in myeloid progenitors or only when the myeloid cells are present at the tumor site, or is it increasingly increased during tumor progression? And what we saw is that this stress response starts in fact in myeloid progenitors. This is what I'm representing here, and it increases during myeloid differentiation. And then when we compared motif accessibility, just reflects opaque chromatin regions. So the transcription factor that NERF2 is just starting to bind, starting to engage a program, we see that motif accessibility start to be accessible in early myeloid progenitors, and this increases along myeloid differentiation. Here I start with myeloid progenitor in the bone marrow, and these are all the different states of this is what I'm describing here: myeloid progenitors, granulocyte monocytic only. Then these are circulating monocytes, and this is 3 different macrophage states in the tumor lesions. And when you look at motif accessibility, you can see that this increases, right? Increases further once the monocyte enter the tumor site. And this downstream gene that is induced by NERF2, which is that stress response, that anti-inflammatory response, also increases along this myeloid differentiation. So start in the bone marrow, and this increases during, increases when this macrophage, this monocyte hit the tumor. Then what we decided to do is to remove this NERF2 from early myeloid progenitors. And we saw a dramatic effect on tumor progression. Well, a significant effect on tumor progression. We then also deleted the regulator of NERF2, this KEAP1, which in fact inhibit NERF2. So when we inhibit KEAP1, we now have more. It's the converse experiment. We have more NERF2, and we saw the opposite effect. And when we looked at the number of macrophages at the tumor, and we saw that this number of macrophages were reduced, and these, in fact, macrophage, there was also a strong reduction of these TREM2 macrophages that I described to you earlier. But within macrophage, we see, in fact, a reprogram, an increased expression of MHC2, of activating molecule. And we saw also increased accumulation of NK cells and CD8 T cells at the tumor site, just by manipulating that NERF2 response. So, and then we treated it with the NERF2 inhibitor, and we are still tweaking the system, and we saw with this NERF2 inhibitor can potentially potentiate PD-1 response in lung cancer lesions in mice. So what we think is happening here, which is something I'm very excited about, is really identify, so is this two-hit hypothesis, is that there are these myeloid progenitors are already sensing, systemic cues that are coming from the tumor, right? Potentially this ROS, that are starting to, in fact, shape myelopoiesis in a pathogenic manner, right? And it's possible that understanding this will enable us to identify novel drugs that really restore healthy myelopoiesis and reduce its ability to be hit, which is going to happen probably at the tumor site, but maybe we reduce susceptibility to additional hit at the tumor site. Okay, so this is the model that we are working on right now. So if I want to summarize now what I described to you earlier, I will describe a different side of myeloid dysfunction that we think we should be focusing on if we want to restore the anti-tumor immunity. We need to identify systemic cues of myeloid dysfunction, targeting pathogenic myelopoiesis as I described to you. There's something I didn't talk about, but we think that the spatial distribution of macrophage, but particularly the dendritic cells at the tumor site, is going to be very important to induce effective antigen-specific T-cell response. But most importantly, what I really described in this lecture is our interest in identifying cellular compartments that are pathogenic and drugging specifically these compartments. So I talked about the TREM2 macrophage, now talking about the IL-4 receptor program. Can we identify and then specifically target those cell types that are responsible for this myeloid suppression, and as much as possible target it to the tumor site? Because if we can do that, right, we will be able to spare the rest of the tissue and really have more flexibility in really reprogramming these myeloid cells in a way that is as immunogenic as possible. Okay, so this is my lab and fantastic Matthew that contributed to the TREM2 stories, and I think I acknowledge many people along the way. And my colleague Brian Brown and Gürkan Moloaglu here, who did the beautiful pooled CRISPR screen.